Alright, hey, how's it going everyone? Mr. Binder here, just taking a look at Chapter 7 of our notebooks or our textbooks. What we did take a look at in Chapter 6 was DNA replication. So basically we're still looking at a cell, an animal cell, and all of these different processes that occur in the cell. So what we're taking a look at now is not how a cell replicates its DNA, but how a cell can take the information that's stored in the DNA and translate that into what's called a protein. All right? Protein, again, is something that we took a look at in Chapter 1. It's a macromolecule, and there's a lot of different functions that proteins serve in the body, but more importantly, in the cell. All right? So throughout this chapter, what we're going to take a look at is how proteins are made in the cell, and it all stems from basically our DNA, or the DNA that's found inside of the cell. We'll take a look at the different processes that occur, some of the different enzymes that are used to convert or to basically translate the information from DNA into protein. And then later on we'll actually take a look at how our I guess genes or DNA can be switched on and off, okay, when specific proteins are needed. So this is the one gene, one polypeptide hypothesis, and basically what this is along our DNA, and again, hopefully you understand that our DNA is organized, well, 3 billion base pairs long, so a total of 6 billion base pairs. That is organized or coiled up and organized in 46 chromosomes, okay? 22 pairs of autosomal chromosomes, and then we have our two sex chromosomes. Okay? So 23 total pairs of chromosomes. All right, and along these chromosomes we see that there are regions of DNA that are specific to a protein. Now remember, proteins can be all sorts of different structures. All right, a good example that I could give you here would be an enzyme. Okay, so if a specific cell needs an enzyme, well, it is going to let the nucleus know or the DNA that, hey, we need a specific enzyme here. Why don't you turn yourself on? Okay, so that gene will become active, and what will happen is the cell will start reading the DNA that has become active, or that specific location in that DNA that has become active. It will then get translated into that specific protein, and now the cell can use that protein for whatever function it is or what it needs to be. So taking a look here, again, DNA located in our chromosomes. Okay, along our chromosomes there are specific genes that code for protein. All right, there's approximately 20,000 genes in our DNA. This number is always up for debate. I think scientists have narrowed it down to about 20,000, so excuse the 5 there. So, you know, let's say approximately 20,000 genes in our DNA. Okay, and again, each gene codes for a specific protein or polypeptide. You'll see these terms as we discuss. The central dogma, okay, Francis Crick, scientist back in the 1950s, basically what he determined was the central dogma for how proteins are synthesized in the cell. And it all starts with our DNA, genes that are located on our DNA. Okay, our cells will transcribe information from genes to essentially messenger RNA. That RNA will then become translated into a polypeptide. And as we've seen in Chapter 1, polypeptides will take specific shape depending on the interactions of our side groups or the R groups there. And again, there are 4 of them. For tertiary folding, we see the hydrogen bonding, okay, the hydrophobic interactions, the disulfide bridges, and the ionic bonding. All right, so taking a look at that, we see that we have our specific protein developing through the central dogma. The overall process, 2 things that we're going to take a look at, transcription and translation. Essentially what happens in transcription, our cell will be transcribing DNA to RNA. Right? And where does this take place? It takes place inside the nucleus. Once RNA has been transcribed from a piece of DNA or from a specific gene, then translation occurs. And translation means interpreting the information found in the RNA and translating it into an actual polypeptide. And this takes place in the cytoplasm as well as the rough endoplasmic reticulum. Some key terms that I want you guys to follow as we do discuss the transcription and translation. Okay, initiation, elongation, termination. We'll see that both in transcription and translation. The TATAB box, introns, exons, mRNA, tRNA, rRNA. There's 3 different types of RNA here, and they all kind of come together to help make a polypeptide. Okay? Translation, ribosome, codon, amino acids, and polypeptide. These are all key terms that I will be using as we do discuss this whole process of protein synthesis. Again, this is just a kind of, let's throw it back to chapter 1 here. We are comparing and contrasting DNA to RNA. Okay, again, DNA is a double-stranded molecule, while RNA is only single-stranded. DNA, adenine pairs with thymine, guanine pairs with cytosine. RNA, when RNA is being synthesized here, especially from a specific region of our DNA, adenine will pair with, well, when RNA is synthesized, uracil will be pairing with adenine in the DNA. Okay, and this is very, very important. So if we were to take a look at our actual example down below here, if the top portion is our template here for our DNA, okay, and the RNA will be synthesized off of the template here, or our DNA strand, our single DNA strand. Well, when an RNA molecule is being synthesized, remember A pairs with T, okay, but when we take a look at if there's an A here in the position of the DNA, instead of a T, which is normally the case for DNA replication, for this mRNA synthesis, okay, a U will take the place of the T. All right, and that makes, that's very, very important, especially when you guys are working on your worksheets through the whole transcription process. Whenever you see an A in the DNA strand, you know that that A will pair with a U in our RNA strand. It's very, very, very important, and you need to understand this concept before you even move further. Okay? So DNA to protein. A protein is made up of amino acid sequences. We know this, right? The subunit of a protein is an amino acid, and there's 20 different amino acids. All right. Well, how does DNA code for a specific amino acid? Again, we have to take a look at, well, how can I, understand someone that speaks French if I don't know what French really means, right? Or if I haven't spoken French before. Well, we need a translator. And how do we get these translators? Well, we can pay a translator, right? But basically, there's someone that's working for both of us so we can understand each other. And again, we see that here with DNA to protein. All right, we have a specific gene, and we need to turn that gene, or the information found on that gene, into a polypeptide sequence, which is made up of amino acids. And how the heck is this going to happen? Well, there's some specific steps that need to come into play here in order for a successful translation, okay, of a gene. So we take a look here. The specific, I guess, how we can say a polypeptide will be synthesized is each amino acid will represent a specific 3-letter code. And this 3-letter code, which is found on our mRNA, and that mRNA will be transcribed from DNA. So we've got our mRNA. Once that, let me write it here, mRNA. This molecule will be transcribed from DNA. Okay? And in this mRNA molecule, we can break this mRNA down into 3-letter codes. And these 3-letter codes stand for a specific codon. Okay, we start from the 5' to 3 order. Okay, so we start at the 5' end. All right, and we work our way down to the 3' end. All right, and we read from the 5' end to the 3' end. All right. Notice this is your chart here, which will basically tell you the different codons that represent an amino acid. I will help you guys to interpret different codons here, okay, and there's specific examples later on. But taking a look here, phenylalanine, this is an example of an amino acid. Again, in your case study, you guys hopefully will research that phenylalanine. It can't be broken down for individuals that have this specific disorder known as PKU. Well, there's 2 different codons, okay, that could be found in mRNA that'll code for a phenylalanine amino acid, and that is UUU. That's one example, and again, the next example will be UUC. Okay. Understand that more than one codon can specify a single amino acid. Okay. And it makes sense because you take a look, 4 to the 3 here, this is the amount of times that I can rearrange our 4 nitrogenous bases. All right, so 4 to the 3, there's 64 different ways that I can arrange the nitrogenous bases here, okay? But if you take a look at it, there's 3 codons that specify stop, okay? And we'll talk about what stop means, all right? So if we take 64 minus 3, there's 61 codons that specify an amino acid. Okay, that's why there's multiple codons for a single amino acid. So here we see that proline has 4. Okay, histidine has 2. And again, we see arginine. Arginine has 2 as well. All right, so it just depends on the codon that you're given, and then having to match it up with its specific amino acid. And this is how I would want you guys to read it. This is the chart that you guys will be given, all right? And what I'd like you guys to do with it is learn how to interpret it. So let's say we start with the DNA here, okay? Oops, sorry. Let's say we start with the DNA here, all right? And the specific 3-letter sequence that's found in the DNA or in the specific region of DNA, which is known as a gene. Remember, our cells are trying to translate the information found in DNA into a functioning protein. So we start with TTA. Okay, well, the first step in trying to understand which amino acid is being translated here, we have to convert this, okay, to mRNA. Okay, and how are we going to do that? Well, what we're going to do is pair up the basically complementary base pairing here, but with regards to RNA. So again, we see TTA here for our DNA. Well, how are we going to transcribe that into mRNA? Well, what pairs with T in DNA? Keeping in mind that we're synthesizing an mRNA molecule here. Well, A does, right? That makes sense. Again, we see T, so the complementary base pair would be A. Here at the end, we see an A, okay? And this is important because it's not T in the mRNA. Remember, RNA does not have thymine as a nitrogenous base. It only has uracil. Okay, uracil takes the place of our thymine here in mRNA. Well, in the DNA sequence, there is an A. What pairs with A when we're synthesizing, or our cells are synthesizing, an RNA molecule? Well, that would be U, uracil. Okay. This is very, very important. This AAU is the codon that will be used to basically put together your polypeptide. This AAU will be the codon that represents a specific amino acid. And looking at the chart to the right here, how can we tell which amino acid the AAU is talking about? Well, we have to know how to interpret the information on this chart here. And essentially what I like to do is I start from the inside and I work my way out. So what's the first letter of this codon? It would be A. So we start here, A. All right, sounds good. Now that we're on the inside, let's work our way out. What's the next letter? A. So we go from A to A. Okay. Now we're on to our second letter. Finally, our third letter. What's the third letter of this codon? U. All right, so we jump to U here. Okay. We jump to U. We have our sequence here, A, U, and that specific codon is asparagine. Okay. So again, you have to use the codon found in the mRNA, and that will give you your specific amino acid. The reason why there's tRNA here and the UUA, this is the anticodon. You do not use the anticodon to interpret which amino acid it belongs to. You actually use the codon, which is found in the mRNA. Please do not get that confused. You will have a totally different polypeptide sequence, and that will not be the specific polypeptide that I'm asking you to code for. Okay, the anticodon is very, very important in terms of pairing up which amino acids should go together. Okay, and we'll definitely be discussing that later on in translation. Let's talk about transcription. Okay, we basically did already. Okay, you guys just have to kind of understand, how it works, or how you need to transcribe the letters from DNA to RNA, and then translate the letters, okay, in mRNA to an amino acid. Okay, the first step though is DNA to RNA. How the heck does an mRNA molecule be synthesized here? Well, it's very, very I'm sure, complex in our cells, but from what we can understand, there's three processes here: initiation, elongation, and termination. Okay, we'll take a look at each of these processes. It occurs inside the nucleus. Okay, DNA, remember, it's found only in the nucleus. I mean, there is some DNA in the mitochondria, but we are not talking about the mitochondrial DNA in this case. We are talking about the specific DNA that's located in the nucleus of a eukaryotic cell. Okay, so three processes that we're going to take a look at: initiation, elongation, and termination. All right, the first step of initiation here. Okay, a couple things that I want to mention. All right, initiation. This is found on DNA. So specifically speaking, our DNA is 3 billion base pairs long. All right, taking a look at which region, all right, or which spot on the DNA that transcription is supposed to occur at, it involves a couple different things here. First of all, we see that there's promoter regions found on our DNA, basically next to each gene. All right, more specifically speaking, this TATA box. Okay, and the TATA box is just a series of TAs, okay, found in the DNA. Okay. All right. So having said that, this box here will help to determine a specific promoter region. All right. The enzyme here that will be synthesizing the mRNA molecule is known as RNA polymerase. All right. RNA polymerase will unwind the DNA as well as synthesize the mRNA molecule. Now, in order for RNA polymerase to do its job, it needs to bind to the promoter region of the DNA. Okay, and specific transcription factors will allow RNA polymerase to bind to this promoter region, or this kind of starting unit here for RNA polymerase to synthesize the mRNA molecule. Okay, the transcription molecule, or the transcription factors, the RNA polymerase, and the TATA box, all of them kind of come together here to form this transcription initiation complex. Okay, and basically it's the area where an mRNA molecule will become synthesized, or the beginning part of it. Okay, the next portion here is RNA polymerase will start to unzip the DNA. Remember, DNA is a double-stranded molecule here, so RNA polymerase needs to unzip the DNA and then start reading the base pairs that go along with the specific strand in which RNA polymerase will be transcribing. And taking a look here, the strand that RNA polymerase will be building this mRNA strand from is not the coding strand. Okay, there's two strands that that DNA basically will be labeled as. Okay, the coding strand and the template strand. Okay, the mRNA transcript will be synthesized by RNA polymerase. RNA polymerase will use the template strand of the DNA in order to synthesize the mRNA molecule. Okay, there are no primers needed here. Okay, again, the transcription factors served as a substrate for RNA polymerase to bind to the DNA strand. Okay, RNA polymerase will synthesize this new mRNA transcript 5' to 3'. Okay, all right. So again, coding strand is not the strand here that will be transcribed. This is not the strand that will serve as the template here for the mRNA molecule to be synthesized. All right, keep that in mind. Well, once the mRNA strand has been synthesized, the last process here in transcription is known as termination. Okay, and basically RNA polymerase will recognize a termination sequence. All right, basically it will read a bunch of A's here. Okay, a lot of A's in the DNA. Well, what happens here is nuclear proteins will bind to a string of UUUUUs on the RNA. All right, once that happens, the mRNA molecule will release from the template strand. Once the mRNA molecule releases from the template strand, RNA polymerase will then release itself from the actual DNA molecule itself. And again, DNA molecule will go back to its normal double helical shape because of the hydrogen bonds that occur. Okay, H bonds. All right, and it's very important here because H bonds are just interactions between molecules here. Okay, and it's very important because again, in protein synthesis, we don't see these SSBs, or single-stranded binding proteins, attaching to the strands and keeping it from annealing. okay, is very, very important. In protein synthesis, we actually just see the strand just anneal right back together. All right, and it's very, very important. Again, taking a look at this picture, we see DNA strands here. We also see what would be, or what I would think would be, mRNA transcripts. Okay? So, again, transcriptions just occurred. We have our mRNA transcript. Where does all this occur? Inside of the nucleus of a cell. Well, before mRNA leaves the nucleus and into the rough endoplasmic reticulum, a couple things need to occur. All right, and these are very, very, very important. All right, because it ensures that the mRNA transcript will actually be translated outside of the nucleus. Now, taking a look, once the mRNA strand has been synthesized, thanks to RNA polymerase, we call this pre-mRNA. All right, again, pre-mRNA has to undergo a couple modifications before it leaves the nucleus. And what are they? This is a test question. It's a huge test question. The poly A tail, the 5' cap, and the removal of introns. Okay? Basically, the poly A tail, okay, what happens here? Well, a bunch of A's are added to the 3' end of the mRNA. Reason why is because it will prevent, or it will protect the RNA from digesting enzymes in the cytoplasm, or cytosol. The 5' cap is 7 Gs. Basically, these 7 Gs act as the initial attachment for the ribosome. Okay, so it can start to translate the mRNA. And again, we see the removal of introns. We have introns here. These introns are just non-coding regions of the mRNA transcript. Basically, they don't serve a purpose for when it comes to translating the actual mRNA and producing a polypeptide. So those introns need to be removed. Again, how does this happen? Well, introns are removed with an enzyme known as spliceosome. Spliceosome will, with the help of SNRPs, kind of act like scissors. Okay, and basically what happens here is these little introns will be removed, all right, and then basically the exons which are left will be spliced back together. Okay. Again, we see the SNRPs here. They bind to the specific regions on the introns. They kind of bring it together like a loop, which is easy for a spliceosome to act on that region and splice it out. Again, this is called basically this whole arrangement, because if spliceosome can cut an exon out, okay, or sorry, an intron out, it can also bring different exons together. All right, and taking a look at this, if this is our pre-mRNA here, all right, and the pink regions of this pre-mRNA are known as introns, again, we have to, or our cell or spliceosome will cut out those introns. Keeping in mind here that exon order is always preserved. So again, saying that these exons cannot be rearranged. You know, this one can't go over here. Okay, and the blue can't be swapped out with the purple. The order of the exons are always preserved here. Okay, but having said that, a specific exon could be snipped out and not used. All right, why would this happen? Well, if there's only roughly 20,000 genes found on our DNA, this alternative splicing method here allows our cells to be able to synthesize up to approximately 100,000 proteins, which is awesome. Okay, so taking a look here, we have our pre-mRNA, and notice the different or the total number of arrangements that this pre-mRNA can take after it has been acted on by spliceosome. Okay, we see 1, 2, 3, 4, 5, 6 different arrangements here. Okay, and each of these arrangements will make for different proteins. All right, alternative splicing, very, very important. It'll be a test question. Okay, so the pre-mRNA had the modifications to it, again, the 5' cap, the poly A tail on the 3' end, and the introns were removed. That mRNA is now ready to leave the nucleus and basically travel its way to the rough endoplasmic reticulum. Okay, now the process here, we don't have a polypeptide yet. All we have is an mRNA transcript. And essentially what needs to occur is the synthesizing of a polypeptide chain here. Now remember, the cool thing about this is on our mRNA transcript, there are 3-letter codons. These codons code for a specific amino acid. So a couple different things need to come together in order for a successful translation of the mRNA into a polypeptide. Okay. So take a look here. We've got a couple different RNA molecules. We've got tRNA and we've also got a ribosome, which is our rRNA. So we have mRNA, we have tRNA, and we have rRNA coming together to help synthesize this polypeptide chain. All right, the tRNA is very, very important here. Okay, again, we have our messenger RNA, which was produced inside the nucleus and then exited the nucleus and is now ready to become translated. Well, another key player here, and it's also another RNA molecule, would be our tRNA, or transfer RNA. What a tRNA molecule brings to the table here, why it's so important, is each tRNA molecule carries a specific amino acid. Okay, carries a specific amino acid. Now, having said that, this tRNA molecule is important. Again, it looks like the shape of a T, but at the bottom here we can see that for each codon that's read on the mRNA, there's an anticodon. All right. Now, are there 61 tRNAs to read 61 codons? Again, this is something that scientists have still yet to prove, but it's a hypothesis out there that there's only 20 tRNA molecules that carry 20 different amino acids. Okay, and the reason for this would be the wobble hypothesis. Okay, now basically what happens is the first 2 nucleotides of a codon for a specific amino acid is always precise. Okay, so here the example is tyrosine. All right, we see that the anticodon here is AUA. Okay, AUA. All right, now again, AU, the first 2 letters here are always specific to tyrosine. It's the last letter that, again, scientists are trying to determine whether or not this wobble hypothesis is occurring. All right, so on the mRNA transcript, we see a C and we see a U. Well, taking that into consideration, this tRNA molecule will pair up with the first two codes of this codon, A and U. A and U. It's the last letter where there is some, I guess, wobble position here that allows the tRNA molecule to understand that it will be this amino acid. Okay, it's called the wobble hypothesis. All right. Now, what I would like you to know is it's known as amino acylation. Basically, once a tRNA comes into contact with a specific codon found on the mRNA, the amino acid will then kind of detach itself from the tRNA molecule, and we see that there will be a growing polypeptide chain. Now, once that tRNA is empty and the amino acid is now attached to the growing polypeptide chain, tRNA needs to go and get another one. It needs to go get another amino acid. So again, amino acylation is the process in which a tRNA and amino acid will come together, and we see an addition of an amino acid here to the tRNA molecule. Okay. So just as I will be asking you a question that what is amino acylation, and what I'd like you guys to say is that, yeah, a tRNA molecule attaches itself to a specific amino acid and brings it back into basically the whole series of events that are occurring to grow this polypeptide chain. It's known as translation, right? Ribosomes. Ribosomes are very important here. Again, we learned in chapter 2 that ribosomes, they make protein. Well, ribosomes just don't make protein. They need a couple other molecules in order for a successful translation or a growing of this polypeptide chain. All right, we see that this molecule here is your tRNA molecule, and we see that this strand here is your mRNA molecule. Okay, this kind of, I guess, beigey molecule here, okay, I'm kind of coloring it. Okay, this is your ribosome. The ribosome's like the house. Okay, the ribosome's like the house. The tRNA is like the guest. Just kind of comes in, enters, stays for a bit, and then it leaves. All right. mRNA, the mRNA transcript here is kind of like the host. All right, it's hosting the tRNA molecule. All right. You'll need to know the 3 different sites here that the R and R ribosome or rRNA here is housing. Okay, the A site, the P site, and the E site. Okay, there's different subunits. There's the small subunit. Okay, our RNA is broken up into the small subunit and the large subunit. All right, these aren't too important to kind of understand what's going on here. What I'd like you to know is that the small subunit it serves as the initial attachment to the mRNA. Okay, and the large subunit will attach itself afterwards. But a couple things need to come into play here in order for that to occur. The translation process, again, the initiation, elongation, and termination, the same terminology used for transcription. Okay, we'll take a look first at initiation, then elongation, and then termination. Initiation. Okay, we've got our mRNA transcript. We've got our 5' cap. Again, what happens here? The 5' cap serves for the initial attachment of the ribosome. And what part of the ribosome? You guessed it, the short, or I guess the small subunit of the ribosome. Okay, the small subunit. Every single polypeptide has a starting codon. It's the same starting codon. What is that codon? AUG. Okay, methionine. So again, this complex, this small unit with the tRNA molecule known as or tRNA molecule carrying this methionine amino acid will attach itself to the 5' cap and scan the mRNA. Okay, it will scan the mRNA. Okay, from 5' to 3' until what? Until that small subunit with the tRNA molecule carrying the methionine amino acid reads the specific codes AUG. When that happens, okay, the large subunit of the ribosome will attach itself, and we are now ready for translation. Okay, the reading frame. And this is very important. A reading frame needs to be established here. If a reading frame is not established, then chances are we won't have or our cell will not have a successful synthesis of a polypeptide. Okay, and who knows at that point what protein will be translated. Okay, we need, or there needs to be structure here. There needs to be a starting point, and that starting point all comes with the specific codon AUG. All right, when I say AUG, you're like, oh, that's the start codon, Mr. Binder. And then when I say, hey, what amino acid is that? You're gonna say, hey, Mr. Binder, that's the methionine amino acid. Yes, methionine is the starting amino acid to every polypeptide chain. Okay, A, U, and G. Once the reading frame has been established, we see that these different sites now are important. We see the A, the P, and the E site. All right, the A site is where the tRNA will eventually where it kind of enters into the large subunit of the ribosome here. Okay, and the ribosome will move from the 5' to 3' position. So the tRNA will then become, okay, if it starts here, it will become the P site as the ribosome moves in this direction here. All right, so the initial transfer of amino acids, okay, occur, all right, from the P site to the A site, and the amino acid transfer, okay, the enzyme here that makes this all happen is called peptidyl transferase. So peptidyl transferase will take the amino acid on the P site, okay, and attach it to the tRNA, which has another amino acid in the A site here. Okay, all right. Again, when the tRNA loses its amino acid, it will then move over to the E site, which serves as the exit site. And again, the tRNA will leave the ribosomal subunit, okay, or the ribosome, and pick up another amino acid, so it can come back and again help to elongate this polypeptide chain. Termination. Well, again, there's a single start codon, AUG. Well, there's 3 stop codons here, UAA, UAG, and UGA. So again, when there's a tRNA molecule reading this UAA, UAG, and UGA, what will happen here is a release factor will bind. Okay, the release factor will then help to release the tRNA from the E site of the ribosome. The polypeptide will then release itself from the tRNA molecule here. The large and small subunits of the ribosome will detach. Okay, the release factor will then obviously remove itself. The mRNA will either be degraded at this point in time in the ribosome or in the rough endoplasmic reticulum, or it might be preserved for another translation of more protein. So. Again, we see here polysomes. Okay, what are the red objects and what are the green objects? Again, this is one huge mRNA transcript here. Okay, and along this mRNA transcript, we see multiple ribosomes reading the information on the mRNA transcript, and the green things here would be your growing polypeptide chains. Again, it's review time. Okay, these are definitely important. You have to know what a gene is. Okay, where is a gene located? What's the main function of a gene? Okay, do we need our genes on all the time? And how do we turn genes on or off? And this is going to bring us to the next section. How do our cells turn genes on and off? Again, if a specific protein is not needed in a cell, then why is the cell making it? All right, and obviously, hopefully, you can maybe connect the dots with where we're going to go in terms of our discussion. When proteins are turned on for whatever reason, and there's an uncontrolled synthesis of this protein, hopefully, well, unfortunately, it's not healthy for the cell. But again, it can lead to different diseases. Okay, and this is something that obviously our body does not want to have. So. Again, please review these terms. Watch these slides as many times as you possibly can. Make sure you're answering the chapter questions that go along with it, and please don't hesitate to ask Mr. Binder for any of the questions or any clarification that went along with this screencast.